Welcome student in the part 2 of chapter 14, that is respiration in plants. In previous video, we had discussed the first step of respiration, that is glycolysis, which is common in aerobic as well as anaerobic organisms. And the product of glycolysis was pyruvic acid. In this video, we will discuss about the anaerobic respiration, which is known as fermentation. Fermentation, which is anaerobic respiration. It is the incomplete oxidation of glucose under anaerobic conditions. It occurs in many prokaryotes and in unicellular eukaryotes. The type of fermentations are alcoholic fermentation and lactic acid fermentation. Let's learn about the alcoholic fermentation. Here, the pyruvic acid, which is formed from glucose, is converted into carbon dioxide and ethanol. The enzymes pyruvic acid decarboxylase and the alcohol dehydrogenase catalyzes this reaction, and the organism example is yeast. Here you can see on your screen that the glucose is converted into pyruvic acid depending on the concentration of oxygen or the type of organism. Pyruvic will either undergo the fermentation or aerobic respiration. The first is alcoholic fermentation. The enzymes that plays important role in alcohol fermentation are pyruvic acid decarboxylase and the other one is alcohol dehydrogenase. The net ATP produced from fermentation of one glucose is only 2 ATPs. Yeast poison themselves to death when the concentration of alcohol reaches about 13% in the medium. If yeast continues to perform anaerobic respiration and if the concentration reaches up to 13%, then that will lead to death of the yeast by itself. The another type of fermentation is lactic acid fermentation. Here pyruvic acid is converted to lactic acid, and examples are some bacteria. Even in some animals, when oxygen is inadequate during, for example, in exercise, pyruvic acid in muscle cells is reduced to lactic acid by the enzyme lactate dehydrogenase. The reducing agent is reoxidized, that is NADH plus H plus is reoxidized to NAD plus in both type of fermentations. But the net gain of both the type of fermentation is too less, that is energy released is only 2 molecules of ATP. There are certain drawbacks of these fermentation. Enzyme production is limited, less than 7%, its energy production is limited, less than 7% of the energy in glucose is released and not all of it is trapped as high energy bonds in ATP. And the second drawback is hazardous products. The products which are formed like lactic acid and alcohol, both are formed which are hazardous products. Now let's move ahead with the aerobic respiration. Aerobic respiration is a complete oxidation of organic substances in presence of oxygen releasing carbon dioxide, water and energy. It occurs in mitochondria. For this, the pyruvic is transported from the cytoplasm into the mitochondria. And for transferring of one molecule of pyruvate, 1 ATP is utilized. So here at the end of glycolysis, we have 2 molecules of pyruvic acid. So total 2 molecules of ATPs are utilized to transport pyruvate from cytoplasm to mitochondria. These are the crucial events that take place in aerobic respiration. Number 1 is complete oxidation of pyruvate by stepwise removal of all the hydrogen atoms, leaving 3 carbon dioxide molecules. It takes place in the matrix of mitochondria. The second event is Passing on of electrons removed as part of hydrogen atoms to molecular oxygen with simultaneous release of ATP, and it occurs on the inner membrane of mitochondria. The pyruvic acid that enters into the mitochondrial matrix will undergo oxidation. In presence of an enzyme, pyruvic dehydrogenase. It requires coenzymes such as NAD+ and coenzyme A. During this process, two NADH molecules are produced from two molecules of pyruvic acid. The acetyl-CoA then enters into the TCA, that is tricarboxylic acid cycle. Thank you for your attention.